Hi everybody, welcome back to TutorMe LS. Apologies for the sound, I'm actually sitting in my classroom right now recording. Today we are looking at DNA profiling, and once again it's going to help our grade 12 life sciences learners pass that exam. What is a DNA profile? It is a pattern of black bars that is left on an X-ray film when an extract of DNA is put through a special biotechnical process. Now, it's a whole lot of scientific terminology here, but it's basically a method of identifying an individual by comparing their DNA profile with another. So to see, this is what a DNA profile looks like, but this is often not what we see in our test exams. You would probably see an image like this, whereby you just have these bars that we can see distinctly. So biological tissue is collected and the DNA profile of that person to whom the tissue belongs is established. So they take that biological tissue, they put it through that biotechnical process, and they get that DNA profile. Now, if the DNA profile of a suspect matches the biological material collected from a crime scene, then that person or that suspect is almost certainly guilty of the crime. And little further in the points, we'll understand why it's almost certainly guilty and not actually guilty. Each pattern obtained is unique to an individual except in identical twins. That is important because identical twins share the exact same genetic material. DNA profiling is a popular method for determining the guilty. However, this method may not always be accepted in court since there is no uniform method for taking the profile. And that's the key thing, why they say it's almost certainly guilty, because there's so many factors that determine whether they are 100% guilty based on the method of DNA profiling. Now, the advantages of DNA profiling. 1, diagnosis of inherited diseases. 2, biological evidence. 3, developing cures of inherited disorders. And the fourth is identification of relatives. Now, under each one, so obviously diagnosis of inherited diseases, so diseases that an unborn or a newborn child has inherited from their parents. The DNA profiling is used for that. Police laboratories use DNA profiles from biological evidence and to track down criminals, and this is what we call forensic science. This is like the CSI sort of things that they do. Developing cures, they look into patterns in large groups of people with and without the disorder, and necessary for designing genetic cures for this disorder. It is also useful in establishing relationships amongst individuals, like, paternity tests, someone that has, was burnt beyond recognition, or someone that was, their remains were left behind. So it really helps in these aspects. However, with the disadvantages, DNA profile is an extremely expensive process. Is often subject to human errors because people are doing this process, they often can make mistakes. It can be used to frame an innocent person, and you'll see this in movies and stuff where they plant biological evidence like hair and stuff like that. Not everybody's everybody's DNA has been profiled, and profiling an innocent person's DNA may infringe on their individual rights. So that's quite a key thing. It's a very touchy subject because you're dealing with biological material. So let's look at an exam tip. Always pay attention to the bar alignment in application questions, which is what we're going to be doing just now. So we're going to work through some application questions, and we're going to see how bar alignment is so important. Also, always know your advantages and disadvantages, as this is often asked in papers. I've seen multiple questions on, these types of in DNA profiling, where they often ask advantages and disadvantages. So let's look at some applications. So here you have a scenario whereby a Detectives, they were investigating a crime scene and they found blood on a broken window. The suspects, they suspected the blood was that of the criminal, and to identify the criminal, they analyzed the DNA sample from the blood and compared it to the 4 suspects. So when you have this is the sample that they collected, and these are the 4 suspects. All we've got to do is we've got to make sure the bars of the sample are identical to the individual. And if we look at it, it's going to be that of Jenny. So they're asking, what is the technique that is used? It's DNA profiling. Who is the possible criminal? It is going to be Jenny. Explain your answer because the bars are matching, or all the bars are matching. Again, see, state one other use of technique. So one other disadvantage, one other advantage and one other disadvantage. So if you look here, I'd say this here is very important to pay attention to because that's basically you need to be able to explain properly how come you are concluding that Jenny is the person, and we say that the profile matches the DNA profile from the crime scene, or that the bars matches the bars in the sample. Second application question. So we have Tom and Maria, and they have 3 children. One of these 3 children are adopted, and they want to see which one is adopted. So remember, with Tom and Maria, with the parents, it is a combination of the genetic material that is passed on to their children. So in other words, their children, which is Anne, Mary, and Steve, have to have a combination of both Tom and Maria. So if we look here, the first one, none of them have it. With Maria, the first one, Anne and Steve have it. With the next bar, Steve has it. With this bar, Anne and Steve. With this bar, no one has it. With this bar, Anne has it. And with this bar, Steve has it. So the only person, which is Mary, does not actually share any, or the bars, none of the bars or the profile is matching. So again, there are no matching bars. So that's the key. When they're asked to explain, there are no matching bars or bands or the DNA profile with both parents. So now we have another application question. Now, this one I particularly like because it's looking at who the possible biological father could be. We have a child and we have a mother, and there's 4 possible fathers. And here it's slightly different because remember, it has to be a combination of both parents and DNA. So the bars need to be a combination of both mother and the father to give us the child. So this is how we would work this. We know for sure that the mother is the mother. So wherever there is a corresponding bar between the child and the mother, we tick off. So that's ticked off, that's ticked off, these are ticked off, ticked off, ticked off. So what's remaining? That's remaining, that's remaining, and that's remaining. So then we go across. If that's remaining, that's there. That's remaining, that's there, and that's remaining, that's there. So the possible father is going to be man 2, which is option B of here, as we can see. So when we have a question like this, make sure we take off the bars that are corresponding between the child and the mother first, and whatever's left behind will be, we need to tick off against the possible fathers. Well, guys, that's it for today. Thank you so much for joining me. Hopefully this has been educational as always. And if it has, please give us a like, subscribe, and hit that bell notification, as well as hype up this channel. It really gives exposure, makes me get out there and do the things that I love doing, which is making life sciences a little bit easier. So until next time, stay curious and keep learning.